So now we have my great friend Vera Gorbunova. You heard from Andrei Salwanov yesterday or the day before. These two are a fantastic team, and they do amazing work, especially on long-lived species, among other things. So over to Vera.
Does the mic work? Okay, good. So thank you very much, Aubrey, for the invitation, and thank you for being so thoughtful to inviting both Andrei and myself. We always enjoyed traveling together, and we enjoying your meetings so much. All right, so let me begin. I should also thank Steve for giving such wonderful introduction, and congratulations on your award. You've been an inspiration for me early in my career, and this is what brought me to comparative biology. Just very quickly before I move into science, I want to say a few words about our journal, Aging Biology. I will not go very much into history of how it came to be. You can read about it on Twitter, but I just want to say that we are doing well. We published our first issue. So these are my dear co-editors-in-chief. And our first issue, so people are submitting papers, and what's different about this journal is that it is all run by scientists and for scientists, and so, don't be afraid, send us our papers. All right, now let's talk about science and comparative biology specifically. So I want to focus on lessons that we learned from long-lived species, because we've been studying them, well, inspired by Steve Austin. We've been studying these species for probably 20 years now, and there are some amazing findings that came through it. Okay, my disclosures first, okay, so as it's already been said, there is dramatic difference in rates of aging and maximum lifespans across mammals. So we want to understand what makes short-lived species different from long-lived, and then to find out what long-lived species do, so that can we use it for human benefit. There are a few species on this slide, and I will not say much about buzz, because Emma will be talking about them, so I don't want to steal your thunder. But bats are amazing. And then I will talk a little bit about naked mole rat, bowhead whale, and then also comparative biology, where we take many species and compare them to each other. And here is the blueprint. So we study those long-lived species, we find specific molecular mechanism, and then we put it in the mouse. Well, mice are good for something. You can use them as test tubes to test those anti-aging interventions that we take from a long-lived animal. If they can help mice, then, well, hopefully they can also help human. It's something that can be exported. And yesterday, Andrei, or even the day before yesterday, Andy gave beautiful talk about naked mole rats. So we've been studying naked mole rats for a very long time. So here is hallmarks of aging picture with different findings from our groups, where we found all these unique adaptations in naked mole rats that address multiple hallmarks of aging. So there is this little rodent that ages extremely slowly, and they evolved various ways of doing it, and some of these ways are more easily translatable to human healthcare than others. And again, back to Andrei's presentation, so we found that hyaluronic acid that is so abundant in naked mole rat is something that can be translated. So it's coming out in Nature next week. It took so long, very long journey. And eventually what we'd like to do is translate it to humans, because now mice can benefit from naked mole rat hyaluronic acid, and now we are starting to test how to use it in people. So this is one successful example of using this pipeline from long-lived animal into human. So now a few words about the whale. So whale, well, as Steve just mentioned, naked mole rat is special because, well, not necessarily it lives longer than human, but it is just very small and it is very unique in its mortality patterns. But if we talk about whale, well, it is actually the only mammal that lives longer than humans. So bowhead whale lives up to 211 years, maybe longer. So that's the only mammal that outlives human by twofold, 2 human lifespans. And they're amazing in many ways. They're also so large. And I could talk about how they avoid cancer, which is another big issue of big animals. You know, how do you avoid tumors? So this is our team in Alaska investigating whales and collecting samples. Again, for the sake of time, I'll go quickly. I must say this story is now in revision for Nature, so we still don't know what the fate will be, but I think the reviewers were kind to us, so I hope we'll manage. So what we demonstrated was that whales don't seem to have more sophisticated tumor suppressive mechanisms. They don't have multiple copies of p53 or RB. So they only need 4 oncogenic hits to develop malignancy, unlike humans that need 5, so which was a big surprise. We expected whales to have additional copies since they're so much larger. And that brought us into looking into DNA repair in whales, because we thought, okay, maybe they just don't let things progress so far. Maybe they repair damage, and this is why they don't mutate their tumor suppressors. And indeed, we found that in the bowhead whale, double-strand break repair is extremely efficient, extremely fast, accurate also. So when we measured mutations in bowhead whales at the site of the damage, we found that they have much fewer mutations, so they're extremely accurate. And here we are comparing them to humans. So human is very hard to beat, but whales are better than us at DNA repair, and they are better than other animals. So here you see mouse. Mouse is the worst, yes, no surprise. They're better than cow, better than humans. So here is another strategy, just very good genome maintenance. And we drilled to the mechanism of it. So we found that in the whale, there is a protein called cold-induced RNA binding protein. It's induced by cold, and this is Arctic whale. It lives in icy waters, so that's probably evolutionary why they upregulated this protein. But now, you know, they have a ton of this protein, 100 times more than humans, and it facilitates DNA repair. So we even found that if we take this protein, the whale version of it, and express it in human cells, we can improve double-strand break repair in human. So isn't it nice? Whale version of KIR protein is not that different from human. There are only 5 amino acid differences, but it is optimized for maximum expression through codon optimization, also through mRNA stability. So they just express it very well. And if we took human cells and put them in cold, we could also stimulate production of KIR, not to the same level as in the whale, but we could stimulate it, and it improved DNA repair. So here are just human cells, and we can see improved DNA repair, which of course led us to speculate about various therapies that are based on cold exposure, brief cold exposure, which is something people been doing for years. In sports medicine, that's how you reduce inflammation from injuries. Cold water swim improves mood, and perhaps it can improve your DNA repair. So this is, again, something we are exploring. Would be very interested to see, you know, all the benefits, because this is a very simple lifestyle modification, and if we can show that it does something beyond psychological effects, so that may be, again, very attractive. Also, yesterday, there was a session on cryotherapy, organ preservation. So I must say that the same protein, KIRB, is actually very important for preservation of organ transplants. If there is not enough of it in the tissue, the organs deteriorate much faster. So this may be another application of the whale protein to benefit human health. Okay, so now we talked about the whale. I want to say a few words about comparative biology, where we're looking at multiple species. So this is the Fountain of Youth. Everybody is familiar with this painting. This is how we see the Fountain of Youth. there are whales, and the closer you are to the fountain, the supposedly longer lived. So here we undertook a study, a comparative study, where we examined like about 30 different species and their transcriptomes, and that part was published in 2022. And right now we are moving it forward, so I will show you some of the new and unpublished data on the proteome analysis. So we analyzed transcriptomes and proteomes across multiple tissues, and we were looking for genes and proteins that correlate with lifespan, either positively or negatively. So here is transcriptome, negatively correlated genes. We found a lot of metabolic genes and genes connected to inflammation and cytosolic DNA sensing, which is, by the way, something that is really knocked down in bats, and they know how to deal with inflammation. But even if we exclude bats from analysis, we still see that these pathways are negatively associated with maximum lifespan. So now what about proteins? So this is all very new. Comparative proteomics is not easy. You have to find paralogs for everything. And also transcriptomics is much more sensitive. So we only primarily looking at abundant proteins. So what we see here is still ribosome, various metabolic enzymes, oxidative phosphorylation, mitochondria. So these pathways are negatively associated with maximum lifespan, even on the protein level. We didn't necessarily see inflammatory molecules, but perhaps because they're below the limit of detection of LSMS mass spectrometry. Now, what about positive genes? That would be becoming even more interesting. From the transcriptome analysis, our favorite part was DNA repair. So we already mentioned DNA repair and how important is it to maintain genome stability, and that was clearly a winner from comparative studies. So to live for a long time, you need good DNA repair. Now, what about proteome? So here we see some new players. We find ECM, collagen, other extracellular matrix components that from the study of naked mole rat, we already learned that ECM is important, but now in comparative sense, we see that ECM is important. Various stress proteins, glutathione, things related to wound healing. And now a really new thing that I'm super excited about are blood microparticles. So we probably you've heard about blood transfusion studies and heterochronic parabiosis when young bloods were bringing beneficial effects. But this is all within species. But here we're looking across species, and long-lived species have higher levels of these blood particles that can carry various types of cargo in them. So we don't yet know what the cargo is. We just know that long-lived species have more of these. So that's something we are actively investigating, and that's actually super exciting, and again, something that didn't come through transcriptome analysis. So this is the summary for that part. Secretory proteins, including blood microparticles, seem to provide us a signal. So now back to DNA repair, a few words about sirtuin-6. So here we went in a more targeted ways because we knew that sirtuin-6 is important sirtuins, it extends lifespan in transgenic mice. But when we analyzed it across species, we found that longer-lived species have more active sirtuin-6. So what does sirtuin-6 do? It's involved in DNA repair, epigenome maintenance, and also suppressing line 1 elements, these genomic parasites that make up major volume of our genome. About 80% of our genome are various repetitive elements. And a couple years ago, we published, together with a group of John Sedivi, we published this series of papers where we showed that line 1 elements can drive inflammation, and that may be another causal mechanism of aging because they get activated with age. So like when we get older, our genome starts to look like that with different parasites waking up. So we need to find out how to suppress them, because once they wake up, they activate innate immune system. and that may be contributing to sterile inflammation and to other age-related diseases. So there is this concept of inflammaging introduced by Claudio Franceschi. So again, this is very new slide. I was really tempted to include it here. So we generated mice with a knockdown of line 1 elements of the most active families of line 1 elements. And these mice show lifespan extension. They also show reduced frailty and reduced inflammation. So this is a strategy to go, and maybe more active SIRT6 in long-lived species. Well, part of the job would be suppressing line 1 elements. Also independently, we can, I know that there is biotech around it just suppressing line 1 elements, but here we can show lifespan extension. So that's super exciting. And finally, the last story I want to show you is about, okay, how do we translate all of this? How we benefit now from SIRT6 for rejuvenation, because, well, we know DNA repair, SIRT6 is more active across species. So this is in collaboration with Steve Horvath and Ken Raj. So the first proof of principle, we took all fibroblasts from old people, like very low passage fibroblasts that Ken Raj sent to us, expressed SIRT6 for 2 weeks, and which was arbitrary time frame, and then measured methylation age. It went down. You can see in majority of our test samples. And it all caused various changes to gene expression related mostly to DNA conformation, chromatin assembly, and so forth. And most excitingly, transposons were silenced. So these genomic parasites were silenced. And they also gained methylation. Because, well, there was a beautiful session on clocks where Vadim and Steve were talking about, you know, the intricacies of the clock. So there is part of the genome that gains methylation with age, and there is part of the genome that loses methylation with age. And if we want to reverse things, we have to perhaps treat those two types differently. So transposons lose methylation, so we want to silence them, and that's part of the thing what SIRT6 is doing, putting them back into heterochromatin jail where they belong. So now another, you know, human factoid. When we were investigating human SIRT6, not cross-species, but in human, in collaboration with Yusin Su in Colombia, we found a centenarian enriched mutation that had separation of function of SIRT6. It had reduced the acetylation activity and enhanced mono-ADP ribosylation. So we started looking then for chemical activators of mono-ADP ribosylation, because that seems to be more important activity if it's enhanced in centenarians. And a very strong activator of this activity comes from seaweed. It's called Fucoiden. It's a polysaccharide. You can see several structures. It's not the same structure because different seaweeds have slightly different Fucoidins. But when we tested, we found that some seaweeds are very strong activators of SIRT6 activity. So yeah, eating seaweed is not going to harm anyone, so it's very safe. And in collaboration with Do Not Age, we decided to do a mouse preclinical study. So we tested different seaweeds. We found one that was a strong activator of SIRT6. And then we started giving it to 14-month-old mice. So mice on seaweed were looking better. You can see just based on the coat. And most importantly, this is frailty score. Mice receiving Fucoiden were having much lower frailty and reduced inflammation, because if SIRT6 reduces inflammation through silencing transposons, for example, reducing DNA repair, that's what we would expect, reduction of inflammation in these mice. That's what we see. And most recent result, survival curve, we even see a signal with lifespan. There was about 7% lifespan extension in these mice that started receiving Fucoiden supplementation fairly late in life, starting at 14 months. So that is all, you know, very exciting. I just want to leave you with these questions. Okay, we know that there are some exceptionally long-lived species, how they manage to do that. We discovered some mechanisms, but there may be many more, and that's a really very fruitful area of investigation. We found, again, from these comparative studies, that it's important to maintain your genome and epigenome very well, SIRT6 is important here, but when SIRT6 we observed that rejuvenating effect of SIRT6. We don't quite understand what kind of template was used to package transposons back into heterochromatin, so we're trying to understand again what kind of template is there, so what information is being read by SIRT6. And I should also say that another approach to it is Yamanaka factors. But with Yamanaka factors, you really rewire everything completely, while SIRT6 is much more conservative. It just reinforces heterochromatin. It doesn't change cell identity. Finally, okay, how do we silence line 1s and restore heterochromatin? Is this sufficient, or do we need to combine different interventions? And this is my final summary of different lessons we learned from long-lived species about genome stability, inflammation, SIRT6 activators, ECM, and of course we would like to, at some point, to combine all of them. So I loved Aubrey's approach into combining interventions, and here we may be able to take all these different adaptations that evolved naturally and then make a super mouse, or well, superhuman at some point, by combining those evolutionary adaptations. All right, so I would like to thank members of my group. Everything was done in collaboration with Andrei Siluanov, and I was showing people's faces as we were going along. So here we are at Cold Spring Harbor meeting. You're welcome to join our group as a postdoc, a graduate student, and we have wonderful external collaborators. Again, I was naming people: Vadim Gladyshev, Steve Horvath, many others. Also our industry collaborators, Oisin Bio, GeneFlow Bio, where we are exploring various therapies with SIRT6 and do not age. And thank you for your attention.
Thanks, Vera. And Vera has very kindly given enough time for a few questions. Over there, George. Wait for the microphone. Wait for the microphone. Where the hell is the microphone?
Here.
All right.
Because I already gave to another one, sorry.
Sorry, I don't know what I just did. What's going on here? All right, George.
Yeah, really interesting. And on the transcriptome versus the proteome, did you look at the correlation between transcripts and proteins in your studies? And did you see anything aging-related?
Yes, thank you for this question. No, we absolutely, we are in the process of doing it. So the analysis is not complete, but sort of the first impression I'm getting is that there are certain transcripts that show good correlations with proteins, and there are others that don't show correlations. So we are trying to sort it out, and we would also like to test some hypothesis whether, for example, if you are long-lived, will you have better correlation or worse? Yeah. So there are lots of questions we want to address, and we also have a metabolomics data set for the same, and then finally we want to integrate all three of them.
Okay, thanks.
Thank you. I have a question. Vera, that was great, and I'm really interested in these blood microparticles that rise or that are elevated in long-lived species. You speculated that they may be delivering some rejuvenative compound. Our company's focused on therapeutic adsorptive particles, so my immediate assumption was maybe they're absorbing some pathogenic compounds, inflammatory cytokines, autoantibodies, who knows what. Can you speculate on that, and also let us know as much as you know about the composition of these particles, dimensions, et cetera?
Yes, no, thank you. This is a possibility, definitely. We don't know for sure what these particles are doing. So we are actually analyzing the composition as of right now. At first, we've just done total proteomics, and that was the signal we obtained in a very unbiased way. But right now, we are purifying them and analyzing. So I don't yet have an answer to your second question, what's the composition exactly, and how does it compare across species, of course. So maybe next time. I know that right now there are several preps on the instrument in Rochester, and yeah, we would like to explore, because yes, they could be sending a signal, they could be cleaning up a signal, they could be just a way of communication between cells. Because again, another speculation, maybe in a short-lived organism, there is just not, the communication may be less sophisticated, and if you want to live for a long time, you just need your different tissues to be much better coordinated with each other. So again, these are all various speculations, and we need to get to the bottom of it.
But I mean, just at a very crude level, these are somehow qualitatively different from exosomes, from extracellular vesicles. They're not like just a type of exosome.
Well, for me also it was fairly new area, because there are all these types of particles. They're classified by size,
right?
Right? And so I think exosome, I mean, they're all particles. I think exosome is a larger one, and here we're seeing the signal for a smaller type of like platelet-derived particle.
I see.
But ultimately, they may be doing similar things.
Anyone else? There's a question there.
Just a question about CRBP1. What do you think it's actually doing? I mean, you do find RNA at double-strand breaks, which are going to be repaired by non-homologous end joining, or is it involved in R-loop resolution, or?
No, but excellent question. I'd love to know that. So there was one paper published on the DNA repair role of CRBP1 using mouse protein. Nobody looked at whale. So what they proposed was that it binds to PAR, which would be similar to RNA, that may be, very similar structure. And then somehow it facilitates recruitment of factors to the double-strand break. So we are exploring it further. Very interestingly, even though the most well-known function of this protein is binding to RNA, which should happen in cytoplasm, where it promotes preferential translation of cold-related transcripts, when we look at normal cells, it's primarily localized in the nucleus. So whatever it's doing to DNA repair is probably its main function, and what it's doing in translation is a secondary one. So my speculation right now is that it is binding, either PAR or RNA, as you said, and probably facilitates phase separation around a repair site. And this is why having more is better. Because if it had an enzymatic function, I wouldn't think they will make 100 times more of this enzyme and things will be, go swimmingly. So most likely it's a phase separator. It has some structural role, but that's yet something we need to explore further.
Yeah, it's very interesting, because if you want to look at, say, Q or DNA PK inside a cell at double-strand break, you need to add RNAs or you can't see it. So.
All right, thanks very much, everybody. I'm afraid we have to move on, but I'm not actually...